Okay, good morning, everyone. Can you hear me?
Yes.
Okay, can you see my screen?
Yes.
Okay. Let me see. I'm just getting organized here. If you don't mind, could you turn on your camera so we can see each other initially, at least? So just so we can introduce ourselves. Let's see. So I think I see 9 people total, including myself. I'm not sure how many people are enrolled. Let me just check real quick. I guess it doesn't matter. Okay, so anyway, I thought we could introduce ourselves first, and I'd like to get a sense of like who you are and what your kind of background you have. But just as a sort of brief introduction, so this is the course at the Graduate Center of Stefan on nucleic acid chemistry and biotechnology. So nucleic acid biotechnology, biotechnology applications that involve nucleic acid synthesis. So my name is Mark Salazar, just you can call me Mark. I have been at the TUM for a couple of years. Before that, I was at the University of Vienna. Actually, I'm still at the University of Vienna. I'm in the chemistry department. So my specialty is within chemistry is nucleic acid chemistry. And so that's my claim to be able to say something intelligent about nucleic acid. So in this course, we'll talk about, I'll start out today with a sort of a background on molecular biology, since the discovery of DNA and RNA, and some basic background about polymerases and transcription and translation, things like that. And then we'll go on to talk about the structure and function of DNA, so sort of like double helix, the details about that, why it forms a double helix, the physical basis for the stability of double helix. For both DNA and RNA, they have somewhat different structures. I'll also talk about the chemical stability, so why RNA, for example, is less stable than DNA, for example. And then I'll talk about the synthesis of DNA and RNA, so the chemical synthesis of nucleic acids. So this is based on an approach that was, well, I'll talk about in detail, but this was an approach that was initially developed beginning in the late 1960s. So it's about 50 some years old, say it's 50 years old. And it's become a critical technology because there's lots of applications for DNA and RNA in modern science. So for PCR primers, for sequencing, for CRISPR-Cas9, for RNA interference. So all of that DNA or RNA is chemically synthesized, almost all of it. And so it's an important technology, and it's important to understand its strengths and limitations. Also talk about so anyway, the kind of chemistry that is used to make it is called phosphoramidite chemistry, and I will talk in detail about that. I probably also mention some alternatives to chemical synthesis. So there's some new approaches that have been that are being developed. I also talk about, and this is sort of closer to my own research, is non-natural nucleobases. So these are essentially non-canonical nucleosides or nucleotides. So these are, nucleic acids other than A, C, G, and T. So, and DNA and RNA. For example, a natural version of that would be, a natural variant would be a 2' methyl RNA, for example, or methyl cytosine, something. There are some natural variants, or the one that's famous now. I'll talk about also the RNA vaccines, pseudouridine and 5-methyl cytosine, that are used in the RNA vaccines and their purpose there and so forth. So there are these series of natural nucleosides, and then there's the common ones, the ACGT in RNA and DNA, and so uracil in the case of RNA, and then there's some variants that are primarily found in, for example, in transfer RNA, and for example, in methylation sites and so forth. I'll talk about that in a little bit detail, and also about sort of engineered synthetic variants. So these are nucleic acids that are made to have specific properties, for example, to resist nuclease degradation. So these are sort of chemical modifications that we can make. Some of them are profound, like we can have tricyclic bases, for example, or totally non-natural bases that are hydrophobic, and we can expand essentially the alphabet from A, C, G, and T to well, the largest alphabet now has 8 bases, for example, instead of 4. Of course, so the 4 of these are totally synthetic, but they have a base pair system. And anyway, so I'll talk about that. And then I'll talk about the applications of these things, particularly in the context of antiviral or antisense therapy, including RNA interference and also CRISPR-Cas9. So as you're all probably aware, CRISPR-Cas9 has become an important technology, but it relies on synthetically made RNA, sort of guide RNA, and we call it RNA, but in practice it does not have to be RNA. And currently the best gRNAs are actually not RNA, but they're sort of combinations, or it's a guide sequence that includes RNA, but also DNA and other chemically modified sugars or bases. I also talked about the chemistry of sequencing and microarray synthesis, and I will probably also talk a little bit about, as I mentioned a minute ago, about, for example, RNA therapy. So, I mean, this has become famous in the past year with the advent of the BioNTech vaccine and the Moderna vaccine that use modified messenger RNA. So they're called RNA vaccines, but they're not RNA. I mean, I guess the proper abbreviation would be mmRNA, so modified messenger RNA. And at the very end, I'll talk about this because this is sort of an important topic these days because it's important to know, what you're getting injected, I guess. And the reasons for these modifications and what they do. So the counterexample of the mmRNA, so the modified messenger RNA, is the one by what's the company called CureVac, I think it was called. It was a partially failed trials of non-modified, so just mRNA. So engineered messenger RNA, but natural in the sense that there's nothing in all of the nucleosides of that therapy were natural, so A, C, G, T, and U exclusively. There may have been some, the full details I'm not sure are known, but anyway, it's basically a natural version, and the ones that work very well were the ones from BioNTech and Moderna, and these have these pseudouridine. I think pseudouridine is the key chemical modification. I mean, pseudouridine is a natural nucleic acid as well, but anyway, but it's not normally found in messenger RNA. Okay, so I'll talk about some of these things, but I thought that before getting started, maybe you could give me some of your own background, and because I can try to address your interest to as much as possible, and also it'd be helpful to understand your background in DNA and RNA and in molecular biology. So I don't know. So at the top of my screen is Debbie. Is that? Can you tell us about yourself?
Okay. Can you hear me?
Yeah. Mm-hmm.
Oh, good, perfect. So yeah, my name is Devi. So I actually just started my PhD this month. So I'm quite new for a PhD student, but I work now in this molecular medicine. So it's more, what is in English? So it's like molecular nutritional medicine. Okay. Yeah. with Professor Klingenspor. So we work more with the brown adipose tissue. And in my work, I would like to know more whether there is some kind of either like transcriptional factors that affect the difference between the brown adipose tissue and then the white adipose tissue. And we would like to try to figure out what is that. So I thought that it might be interesting for me to maybe know more about the nucleic acid basic something, and then it might be also in the future I might also work with also CRISPR-Cas9. So okay. It's nice.
So by transcription factors, you mean these are like proteins that bind to DNA and initiate transcription. Okay, so I do have some history in DNA binding proteins. I mean, we don't really do that much, but some of the key players I know that they essentially develop a lot of the technology for detecting this, so sequence specific binding. There are several approaches that were developed. I can, if you remind me later on, I can try to talk about these. So when I get to towards the end of the class, so whatever in 8 weeks or so, maybe remind me. I can talk about that in a little bit more detail. Some of the experimental approaches that have been developed to look at the sequence specific specificity of DNA binding proteins. So right now I'm working on RNA binding. But because the DNA binding is more or less well understood. I mean, not at least the approaches have been developed. But what I'm working on now is approaches to do similar things, but for RNA binding proteins. Let me close my window. Sorry, there's a garbage truck driving by. Okay, well, thank you, Rose. And how about you, Pia?
Yeah, my name is Pia. I started my PhD in last December, and I'm working on a project to investigate the communication of two plant beneficial fungi and the poplar. And we will focus on the communication through volatile organic compounds, and we want to investigate the molecular mechanisms that are involved in those interactions. And since I did my master studies with focus on microbiology, I would like to refresh and expand my knowledge in nucleic acid chemistry.
Okay. And have you done any experiments with nucleic acid, like PCR or sequencing or anything? Yeah. You have? Okay.
So I would say I have basic knowledge and.
Okay. Okay, so I am not a molecular biologist. I mean, I sometimes try to, write manuscripts in that direction. But I mean, my background is actually in physics and physical chemistry. And in the past 10 years, I sort of tried to switch over to nucleic acid chemistry, but it comes from a relatively, from a chemical background rather than from biology. Of course, you can't really do research on nucleic acids without, some pretty detailed understanding about how it fits into biology. But certainly that's not my strength. But anyway, hopefully I can say things that you don't know already. Okay, how about you, Sabrina?
Can you hear me?
Yeah.
I'm Sabrina and I'm a PhD student too. I have passed my first year, and I have a look for some courses which I would like to do. And this course I really want because I have the study of molecular biotechnology, and I think it is in every time good to go back to the rules, and sometimes I get some new stuff, and I hope for it in this course.
Okay, what is your PhD? What area?
In the area Institute of Pharmacology and Toxicology.
Okay.
But I can't really talk about my PhD work, and so I'm very sorry.
No, it's no problem. Is there any particular area within sort of nucleic acid chemistry that you're interested in?
Very important for me is some new cases of detection for some nucleic acid, and in application, in some kind, what can I do, what I don't know till this time now.
Okay, so I did, it's not on this list here, but I do have an hour worth or something about the detection of nucleic acid, so fluorescent and radio labeling and things like that, and so hopefully that will be useful. But anyway, let me know if there's something that you think is missing, and then I can try to address that as well. Okay, how about you, Karina?
Yeah, my name is Karina Stoll. I'm a food chemist, and I work, or my PhD started in December 2019. I work in the group for cereal research. So my PhD topic is about wheat malt. I want to use it in the bakery. And for that, I want to know which parts are important. So the enzymes or the matrix components. And to find out more about this, it is useful for me to use some inhibitors for special enzymes. And to get them, I started to clone several inhibitors from wheat to E. coli, so like ligase-independent cloning, PCR, so the basics of this part, I would say.
Okay, so I don't know I wish I knew more about cloning and expression of proteins and E. coli, stuff like that. It seems like extremely useful skill. But it's not something I know a whole lot about. But anyway, hopefully you'll find some aspects of this useful. So there are some things that maybe in that direction, perhaps chemical modifications that improve transcription or translation, things like that perhaps could be useful. But you're in Katharina Scharf's group?
No, it's in the chair of Professor Becker, the Brewing and Beverage Technology, the small group.
Okay, because I don't know if you know her, but she's also in sort of cereal, but she's not here anymore. I guess you know that. Okay, thank you very much. How about you, Wendy?
Hello, my name is Wendy, and my background is bioinformatics. So my research is focused on analysis of the data from the database or from hospital, and also I develop machine learning method. And my research focus on human cancer. So that's why I want to know more about the nucleic acid chemistry more deeply. Yeah.
Okay. So I won't do anything related to bioinformatics, but anyway, because it's almost a different topic. But hopefully, I think it's probably important for people who do bioinformatics related to especially nucleic acids.
Yeah.
You have some understanding on what they're actually.
Yeah.
Okay. How about you, Oliver?
Hi. So my name is Oliver. I did my bachelor's and master's degree in brewing and beverage technology here in Weinstefan. I'm working at the research center Weinstefan for food and beverage, for brewing and food quality. It is complicated name. We're on top of the Weinstefan mountains. So if you ever walked up to the brewery or the beer garden, you walk past our building. It's not very beautiful, so you may not have noticed that there is something in these buildings. I work a lot, or this institute does a lot of microbiology for the industry and research on it. So food pathogens, food spoiling organisms, and detection and characterization of these. For my thesis, I work a lot with, on the one side, MALDI-TOF identification of microorganisms, but that doesn't work for everything. So a lot of PCR sequencing with the standards of 16S, 26S, ITS for fungal identification. I also want to include a lot of next generation sequencing, metagenomics, and so on. I've also got a research project going on exploring some alternatives to the traditional PCR methods for these cases, so for microbial identification, which also includes a lot of primer design, and nanopore sequencing will be employed. And I'm also really keen on employing nanopore sequencing for metagenomics. So yeah, with brewing, I unfortunately only have the basic genetic and biochemistry lectures. I've got a good teacher in primers and PCR with Dr. Hutzler. I'm not sure if you know him. But I'm keen on reworking my basics on DNR chemistry, and also hopefully we'll get to some, well, I'll get some insights into why my primers sometimes may not work, or get better ideas on how to improve my reactions. So yeah.
Okay. Yeah. So I generally don't talk about PCR in any detail, or maybe nothing, because these days almost everybody has done it and probably knows just as much about, you know, the sort of the practice of PCR. I think probably the overlap with what I'll talk about is sort of the sort of physical chemistry of DNA, sort of what kind of factors go into predicting melting temperatures, stability, and things like that. Because those, in the end, well, these are just, you know, predictions that are based on averages of, you know, relatively small numbers of experiments. So we can predict melting temperature, or we can calculate melting temperatures, but these play a big role in PCR, but it's a fairly imprecise science. So, you know, the sequence space of DNA is astronomical. So there's, you know, trillions of different possible primers. And, you know, the calculations, the experiments that go into measuring or predicting or modeling the melting temperature of a 20-mer, you know, are based on 10 DNA sequences or something like that. So you're going from like 10 sequences to whatever trillions. And so you're going to miss a lot. And I'll talk a fair amount about various approaches and fine-tuning approaches, you know, based on salt content, based on all sorts of things. But in the end, these are just sort of estimates of the melting temperature. But anyway, so hopefully at least that part will be useful. Okay, how about you, Josef? We met, I think, last semester as well. Yeah. Mm-hmm. Mm-hmm. Okay. 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 So yeah, so I won't talk about those things and too specifically, but hopefully some of the other stuff will be related. Okay, how about you, Pari?
Hi, can you hear me?
Yeah. Mm-hmm.
Okay, great. So my name is Perry, and I'm an external PhD student in TUM. Our research is in neurobiology, and I'm working on CRISPR-Cas9 cell lines, that they are reporter cell lines, and I'm doing a differentiation on this iPSCs to dopaminergic neurons, and doing some experiments on them. So yeah, as I said, I'm working with this CRISPR-Cas cell lines, and I'm doing a lot of
Okay, so there's some noise. I'm not sure where it's coming from.
Sorry, yeah, that's my colleague. Yes.
Okay, no problem. No problem. So in the past, I haven't talked too much about CRISPR-Cas9, but I'm writing a grant on it, so I know a little bit more. And so I will try. I'm writing a grant on large throughput approaches to determine the the optimal chemical modifications for in the design of guide RNA. So anyway, so maybe I can talk a little bit about that if there's time. Okay, great. And how about you? I think your is Layla is your pronunciation of your name?
Yeah, Layla.
Okay, we can. Can you hear?
Can you hear me now?
Yeah.
Okay, sorry. Hi everyone, my name is Layla. I'm a first year PhD student here at the chair of human biology. Is it more clinical? I investigate the effect of various proteases on the mucus plexus. So I get patient samples from Germany, Hungary, or all over Europe, patients who have irritable bowel syndrome and different subtypes, and then I test them on submucous nerve. This affects, long-term activation. My master's is also in anatomy, so everything I know is very gross and very on a larger scale. I took my chemistry and biotech classes while I was an undergrad, but that feels like ages ago. So I'm looking forward to refresh my knowledge on this. I know basics of DNA and RNA and transcription translation, but I'm more interested in the applications of it. So.
Okay. And you have a very nice English accent. Where are you from?
I was born in Bosnia, but I grew up in America. So.
Okay. Okay. Where in particular?
I went to the University of Maryland for my bachelor's.
Okay.
Yeah.
Okay. I was born in Cincinnati, so I.
Oh, okay. Complete other side. Not complete other side, but.
Not too far. I mean,
yeah.
These things go. Okay. How about you, Lisa? Are you there? Okay, Lisa may not be here. Anyway, okay, so let me just talk about some sort of mechanics of this. Okay, so this is my email, but you have it already. You have, I think, an email from my Leibniz email, but anyway, it's the same account. So we'll meet here about roughly 10 times for about an hour and a half. Some rescheduling will almost certainly be necessary because I can't make it every Tuesday. So we will probably, I'll try to give you, at least a week warning when that happens. And I don't want to open my calendar to right now, but some of those, probably 1 or 2 of those will have to be rescheduled to some other day. So we can either extend by 2 weeks and stay on Tuesday, or we can just do it some other day of the week. So this is for the Graduate Center, the Weinstein Graduate Center. And so what they told me is that I should set some sort of reasonable attendance standard because you get a certificate. Now, I don't know how important the certificate is to you, or if it's, it's useful or important, or is there some usefulness to getting the certificate from the Graduate Center? Is it something you want to get?
Yes, actually, so in order to get graduated, you need to complete 6 SBS, I think, of the the courses and the seminars. So
okay.
Somehow, so, for some people it is necessary, and so some people they just do it over.
Okay, but this sort of counts towards a normal TUM course requirement. Okay. Okay. So anyway, so I mean, probably if you don't care, let me know, just because then I won't worry about it, but I won't keep track for you, and then I won't anyway. But so anyway, what I had in mind is that have a minimum attendance of 70% and participation. So I will give a few homeworks. And I generally don't check them, but I just sort of check that you sort of made some sort of decent effort to do it. This sort of basically on the sort of everything is on the Rna system. And I guess just to keep rough, I mean, so Zoom keeps track of attendance automatically. If you go to the settings, you can see exactly when everybody logged in, logged out, and so forth. But anyway, I'm not trying to. I don't want that sort of. It's not easy to do. So it would be great if you could just send me an email after each class saying, I was here, I was there, just so, and then I can keep track. If you can't be here, let me know. And if you don't think you can make to the 70% attendance and you want to still get the certificate, let me know, and then we can find some sort of option, like do an extra homework or something like that, or I don't know, something easy and generally painless. But anyway, we're all adults here. I don't want to keep track of you. We should be doing these things just because we're interested in them. But anyways, still I need to report to the Graduate Center who participated. So anyway, I do need to do some basic record keeping. Okay, so here's maybe a more detailed overview. So today we just sort of talking a little bit. For the first half an hour, I'll talk about discovery of DNA, transcription, translation. That's very basic stuff that you probably all already know better than I do, but anyway, just in case, feel free to correct me. And then we will transition either late today or tomorrow onto the sort of structure of DNA and RNA and chemical stability, thermodynamic stability, base pairing, hybridization, effect of melting and mismatch. So melting, what affects melting, what mismatches do to the stability of DNA and RNA. And then in the weeks 4 or 5, roughly speaking, I'll go into more detail about the chemical synthesis of DNA and RNA. So it's not a super small topic, but it's probably enough to fill 2 or 3 hours to give you a sort of basic understanding on how it works and what its limitations are, and maybe some of the sort of directions in which people are moving to sort of make it better, and to be able to generate DNA and RNA more efficiently. So the main thing is that DNA and RNA in cells is really pretty long. So DNA is whatever, very, very, very long. And RNA is generally pretty short, but it can be quite long. Like messenger RNA is typically a few thousand bases long. And it's not possible to chemically synthesize something that long. So there's various approaches. One is to sort of make short segments and then sort of attach them together. But sort of the holy grail of chemical synthesis would be to be able to extend synthesis to beyond the current limit. So for DNA, the current limit is probably on the order of 200 nucleotides in length, and for RNA, it's maybe 50 or something like that. And so this is pretty short by natural standards, and those you can ligate things together. There's lots of things you can do, but we can't really compete with nature, and so there's still some effort going into trying to do that better. Okay, so then, as I mentioned, I'll talk about these non-natural sort of engineered modifications and their applications. So these are modifications to the backbone. For example, you can replace the one or both of the oxygens on the backbone with sulfur, or other things as well. Sulfur is the most common modification. So essentially you oxidize with sulfur instead of oxygen. And then you get increased stability, just because enzymes don't completely recognize creates a subtle structural modification that then slows down enzymes that degrade DNA and RNA. But you can also change the sugar. And we'll go into detail about the types of modifications you can make to the sugar. For example, you can replace the hydrogen. You can replace the hydroxyl group in RNA with a fluorine molecule, for example. It's called fluoroRNA, and that's a very common modification. And that's something that gives you something that's RNA-like, but much more chemically and enzymatically stable. So I'll talk about some of these applications. Then I will probably spend one day talking about fluorescence and generally labeling of nucleic acids. So like radio labeling too. Radio labeling is really not that common anymore. So I'll just sort of talk about some of the basics. But fluorescent labeling is pretty complex. There's lots that can go wrong in fluorescence labeling, and it's also the basis for most nucleic acid detection and technology. So PCR is typically based on fluorescence and sequencing as well. So Illumina sequencers use fluorescence. But anyway, there's lots of issues related to avoiding artifacts in fluorescent-based measurements. So now I'll talk about microarray synthesis. So microarray synthesis is sort of, it has been largely replaced by sequencing, but there's still lots of applications where DNA microarrays are widely used, for example, in genotyping. So these days, if you send off your DNA to figure out, what your ethnicity is or who your relatives are, they typically run a genotyping array. So it's a DNA microarray, which is hybridized with DNA that's extracted from you that you give to them. And the reason for that is because, you can do the same thing with sequencing, but sequencing is at least an order of magnitude slower and more expensive. So DNA microarrays do some things that sequencing doesn't do, and they're also far faster and cheaper. So for things that you really don't need to know the identity of every nucleobase, microarrays are a good alternative. And so one of my main research topics is exploring alternative applications for microarray technology. So for example, we do large-scale synthesis of libraries, of DNA libraries, using microarray synthesis. So for example, for information storage, so you can store digital information in DNA using that approach. So I'll try to address some of these topics. Also talking about the chemistry of sequencing. So, sequencing is a chemical process. So it involves using modified nucleic acids, typically with a reversible fluorescent label and reversible terminators, and there's some sort of interesting chemistry associated with that. So essentially with the exception of for nanopore sequencing, which is sort of a direct sequencing approach, like Illumina sequencing or any of the other ones which sort of have lost favor, like sequencing by ligation, for example. Those are all sort of involve chemical modifications. So I'll talk about some of those. And then at the very end, the last day, I'll talk about some new applications and developments in nucleic acid chemistry. So hopefully I can say something about CRISPR-Cas9 and how that works. Probably most of you have a sense already. But I'll also try to talk about some of the applications of engineered nucleic acids in the context of CRISPR-Cas9, essentially to improve the efficiency and reduce off-target effects by chemically modifying the guide RNA to be more specific and to be more efficient, essentially. Okay. Feel free to interrupt me or correct me. And as I talk about this. Anyway, so let me just give you a sort of a quick background on the history and prehistory of nucleic acids. So I guess I chose this slide. Of course, the real history goes back, to the beginning of life, but sort of our understanding of the function of DNA goes back to Wallace and Darwin and Wallace. So they introduced the theory of natural selection. So the idea is that members of a population who are better adapted survive and they pass on their traits. Of course, at the time they didn't know what the mechanism for passing on those traits was, but clearly it became clear with Darwin that there had to be such a mechanism. There was some information that was present in organisms, and that information led to a series of traits of that organism, and that information was somehow passed on to the next generation, which led to the same traits occurring in the next generation. But they had no idea what that transmission of information, how that transmission of information worked. So then in about a decade later, Gregor Mendel in I guess Austria, or at least the Austro-Hungarian Empire, he published his investigations on the inheritance of genes. So they're called alleles in pea plants, and you've all seen these experiments with peas, with wrinkly peas and yellow peas and green peas. And he sort of worked out some basic sort of rules of thumb for how genes were transmitted. Again, so that they were sort of forming some sort of idea as to how this information got mixed in sexual, sexually reproducing organisms. So, you pass on some information from one parent and some information from another parent, and this information gets mixed in some way. And so he would start to elucidate, that sort of mixing mechanism of information from the two parents. So in 1869, Friedrich Nietzsche, he discovered what he called nuclein. So this is a weak acid in the nuclei of white blood cells. So this is what we now call DNA. So all he knew at the time was that there is this weakly acidic substance in the nuclei of these cells. So in 1910, Phoebus Divine, he discovered ribose and deoxyribose and showed that DNA contained the 4 canonical bases that we know. So adenine, guanine, thymine, and cytosine. And so these are the nucleobases, right? So this is the nucleobases, and then there's also a sugar, and then there's also a backbone. So DNA has these 4 components that sort of, well, of course, they're all connected, but we divide DNA into, since the 3 basic components, the backbone, which is this, this phosphate chain, and then the sugar, which is in either deoxyribose or ribose, and then we have this connection, this glycosidic bond to a sugar, which is either a pyridine or pyridine, and we'll get back to the structure later on. But anyway, he found out that there was approximately equal, or as far as he could tell, an equal amount of adenine, guanine, thymine, and cytosine in the cells. And so he proposed the Tetranucleotide Hypothesis, and we'll come back to this in a few minutes in a sort of a brief exercise. And so this is sort of the proposed structure that you can see here, and this sort of makes big sense at the time, that if you have roughly these four molecules, they're very similar to each other, and they're present in very similar amounts, then they maybe form a molecule such as this. There was no idea at the time that this type of molecule or these building blocks had any role in storing information, because there's no really, this is sort of an independent experiment. You just discover that there's these 4 molecules in cells, but you don't really know what they're doing there. It's just sort of interesting that there's equal amounts of all 4 of them. Of course, now we know that they're not exactly equal, but we'll come back to that. So then in 1929, Friedrich Griffith, he discovered that hereditary material from dead bacteria can be incorporated into live bacteria and change that bacteria. And so this was an indication that there is some sort of a substance, a molecule or whatever, that is sort of independent of the cell, right? That sort of can be taken out of the cell, it can be moved around. And of course, this all makes sense, right? Because we know that that cells pass on this information to other cells, their daughter cells. So we know that there's some sort of chemical or substance that is transferred between cells and that transmits the information content of those cells to the next generation, or between, in the case of bacteria, between not necessarily related cells. But anyway, he didn't know what that hereditary material was. So then in 1941, Beadle and Tatum, they used X-rays to cause mutations. And they showed that this stuff, these genes, this whatever it was, whatever molecule it was, could be damaged. And you would then see that damage by formation of misformed proteins, for example. So you're essentially mutating the organism by passing X-rays through it. So that suggested that for every gene there is one enzyme or protein. Yeah. So this is the sort of the beginning of the central dogma of genetics. And there's some indication. I mean, this is nothing super. I mean, especially from our perspective now, it makes sense that every gene would lead to one protein. But it's good to confirm that. So clearly, if you're having information transfer, you need some sort of an entity which has information for every single gene, and that information somehow gets transferred from one cell to the next. So 1948, Avery, McLeod, and McCarty, they separated a virulent strain of pneumonia into their macromolecular fractions, so RNA, DNA, and protein fractions. They didn't know exactly what these fractions, the molecular details of what these fractions were. But they found that they could transfer the properties of this initial bacteria only via the DNA fraction. So this was an indication that this particular fraction was the one that was containing the information. Of course, we know now that, of course, in some ways, all the information is contained in all three of those components, right? So the DNA contains information, but the RNA contains that information too, at least to some extent, right? I mean, like ribozymes and ribosomes, for example, don't contain, or whatever, non-coding RNA may not contain critical information. But the DNA transfers information to the RNA. So in principle, the RNA has that information as well. And of course, the proteins have that information as well, but not in a readable or not in a currently useful format for us. Excuse me. So in 1915, Erwin Schargraf, he discovered that the previous hypothesis that there is a 1-to-1 relationship between the 4 nucleobases. That was incorrect. And it's actually a 1-to-1 ratio of adenine to thymine and guanine to cytosine in DNA. So this was sort of here it says that it's not a disproval of the Levine's tetranucleotide hypothesis, but it's sort of a refinement, actually, right? So because in most organisms, humans included, it's pretty close to a 1-to-1 ratio of all the 4 bases, but it's not exactly that. So all organisms or all species have a slightly different ratio, AT to GC ratio, and it's close to 1-to-1, but it's not exactly. But anyway, this is certainly important information. So in 1952, Hershey and Chase, they proved that DNA and not protein stores genetic information by tracing radioactive, so P32 labeled DNA and S35 labeled protein from bacteriophages infecting E. coli. So bacteriophages are viruses that infect E. coli, and they radio-labeled the protein components of the bacteriophages and with sulfur and the DNA components or nucleic acid component. I guess it must be a DNA virus, but anyway, so with P32, and then when the virus infected the bacteria, only the S35 label, the P32 label part was transmitted, indicating that the DNA is storing the genetic information. So this is a slightly more detailed look at that. So again, the idea is that you have this bacteriophages, and they have this protein component, sort of scary looking protein component, and they store some DNA in the head, and then they inject that into bacteria. And so what they did is they labeled with sulfur the protein part, the shell part, and then the DNA part was labeled with phosphorus 32. And they found that after infection, there was no sulfur labeling entering the bacteria, which is sort of the definitive proof that the protein content does not transmit information, and it has to be the DNA or RNA, I guess, in the case of but anyway, I think they didn't know that at the time that they're also that it's both RNA and DNA viruses. And then 1951, these famous experiments that was based on the crystal structure of Gosselin and so Wilkins, Franklin, Franklin, and they did a X-ray diffraction pattern of DNA and found this double helical structure. Of course, they didn't know much more than that. But anyway, by then it was pretty clear that DNA was storing information, and the DNA had this double helical structure, and that the A to T ratio was 1-to-1, and then the G to C ratio was 1-to-1. And so this was basically enough information to sort of start putting everything together and to sort of hypothesize that DNA stored information and transmitted information between generations, and that the structure of this molecule had these, excuse me, had these 4 nucleobases, and that the A and T were somehow an equal ratio, and the G and C were an equal ratio. And then they had this this helical structure of DNA. And so that, of course, led to the hypothesis by Watson and Crick that so they knew back from the tetranucleotide hypothesis, they knew the molecular structure of DNA. So they knew that there was a sugar, they knew that there was this phosphate backbone that connected them, and they knew that there were these 4 nucleobases. And anyway, so Watson and Crick were able to sort of pull this all together and come up with a hypothesis for the structure of DNA, and they turned out to be right. So again, the structure is shown here, and so they won the Nobel Prize for that a few years later. So just a side note about her. Rosalind Franklin. So she did not win the Nobel Prize, but she probably, if she had lived, she probably at least would have deserved it. But she made some really critical, even though she died very young, she made some really critical discoveries, and she might have been, won even 2 Nobel Prizes like Marie Curie, because she was also involved in some other key discoveries that were also awarded Nobel Prizes. So had she lived longer, she probably at least would have deserved 2 Nobel Prizes. Okay, so I thought we could do a short exercise, but let me send it to you by email so you can, and we'll take like 15 minutes to do that. Let me just send it to you if I can. Just give me a second to figure out how to close this screen. Okay, I can show you while I'm sending it to you by email. I can let you, so the idea here, I can sort of introduce this. So you can see it, right? So the idea here is that, so Phoebus Levine, he formulated this tetranucleotide hypothesis. And, it's not that far from being correct, right? So this looks a lot like DNA. You have the sugars, the nucleobases, the phosphate backbones. But if you sort of write down the structure, it doesn't look like you'd store information with this tetranucleotide. But you can, in fact, store information in the tetranucleotide. And so the exercise is to determine how much information you can store in the tetranucleotide, and whether that's sufficient information to encode for the amino acids that we know we need. So is there enough information? Can you store enough information in a tetranucleotide to be a viable way to store DNA? So let me send you the exercise. Let me just figure out your emails quickly. Okay, so you should get it in a minute. So just take like 10 minutes or something like that to go through this, and then we'll discuss it once you've made some progress. Let me know if you don't get it. You should get it within a few seconds.
Just to confirm, so this for task A means that all 4 nucleotides have to be contained in these. So no tetranucleotides of, let's say, 4 times adenine.
Right. So we're preserving the tetranucleotide in the sense that there's this 1 to 1 to 1 to 1 ratio.
Yes.
And we're just sort of rearranging things, basically.
Yeah. Okay. Just wanted to confirm.
Okay. Did you get the email from me? Okay, so that has the same thing, but maybe it's easier to read. Is it clear what I haven't talked about the 3' to 5' linkages? Is that clear to you what that means? So that just refers to the sugar, right? So the 5' carbon is the one that's at the top, right? So the one that has 2 carbons next to each other, attaching to the phosphate, and the other So here I can, yeah, so where my mouse is, this is the 5' carbon here, and this is the 3' carbon. So you can also reverse the direction of things. You know, so you can have 5' to 5' linkages, theoretically. And then you can have sort of increase the chemical diversity by allowing 5' to 5' linkages. So normally it's 3' to 5' linkages only. But in principle, you can have, you know, chemically you can have 3' linkages and 5' linkages, which then increases the number of combinations that are possible. Okay, why don't we take a look, start taking a look at it. Have you had enough time to at least think about it? Okay. So, okay, the first one is sort of easy, is relatively easy. So again, we want to retain the 5' to 3' linkages. So we'll just represent that by an arrow. So the convention in nucleic acid everything is to always refer to sequences 5' to 3'. So you see, and we'll always retain that convention. And if you ever see a DNA or RNA sequence written down, whatever, A, T, C, G, say, you have to always assume, unless it's specified otherwise, that this is the 5' side and this is the 3' side. So let me just sort of get that out of the way. And if it's ATCG, but this is the 5' side and this is the 3' side, that's a different molecule. So just make sure you keep in mind that unless specified, we're referring to 5' to 3', and that it makes a difference. So they're two totally different molecules. Okay, but let's call this arrow 5' to 3'. And then so we can just sort of abbreviate one structure as A, C, sorry, that should be a C. Okay, let's keep it as a G. C, T. So it's a little bit hard to write using the mouse here. Anyway, so that would be sort of a simple way of symbolizing each of these structures. So then you can say, okay, we just always keep, since we're invariant in rotation, doesn't matter. We just keep the A in the same position and say that can also be a C and so forth. So how many of these structures are there? Anyone?
I think 3.
Sorry, 3?
Yes, is that right?
Okay, I think it's more than that. Anybody else? Oops. Okay, so my PowerPoint just crashed. Let me. Oh, there, it's back.
So first I get to 6.
Okay, hold on a second. My PowerPoint just crashed.
Okay.
So let me restart that. Okay, I guess then didn't like the drawing. Let's see. Okay. Okay, so 3. Anybody else have a different number?
So first I thought it would be 6, but then
Okay.
because I didn't have the arrows, I thought that some of them are the same. That's why I crossed out 3 of them. So I think it's 6.
So I would say 6. Anybody else? So let me try to write this. See hopefully the program doesn't crash again. So A, C, G, T. Let's keep the arrows always going clockwise. So then A, C, T, G. Then, so we've run out of the, starting with A, C, so we go to A, G. And then C, T, and A, G, TC. And then I think then there's just two more. So A, T, CG, and A, T, GC. Right? Does it seem like a reasonable way to do this? So the arrows are always the same in this case. So I think this is correct, but anyway, let me know. Those of you in bioinformatics, your brain thinks a little bit more rationally in this kind of thing. Maybe you can help out. Anyway, so I think there's 6. Yeah, so these are all chemically distinct structures because this represents a chemical bond that has a directionality. It's not like a, just like a carbon-carbon bond. You can flip it around and it's still a carbon-carbon bond. But because these molecules have a structure, so you have what is, so the structure I can just, okay, it's shown here, but so there's a carbon there, which is the 5' carbon. I'll talk about the naming system later on. And then this one down here is the 3' carbon. So the one that attaches to the oxygen there is the 3' carbon. One that attaches to the oxygen up here is the 5' carbon. And so if you reverse it, you get a different molecule, right? And so that takes us to the next one, where we can now change the direction of the arrows, right? So let's see. We start out with ACGT. So the original molecule, or the original directions of the bonds. So again, the arrow represents 5' to 3'. So let's just stay first with this particular orientation. So ACGT. So we keep the order ACGT, and then just reverse bonds, right? So then, for example, we can have A C. Sorry, that's a C. Can I undo that? Yeah, okay. G, T. Okay, so the question is, does this work molecularly? Yeah. So here we have does that work at all even? So this would imply that let's see if I can draw. I don't know if I can draw the structure that fast, but this C has a 5. If we stretch this out, it would be no, we can't even stretch it out. So well, I guess the issue is if we associate each sugar with a direction, right? Because so these have sort of an arrow associated with them, which is, say, points to the 5' carbon. Well, that's a different convention that we used before. Says points to the 3' carbon. Then this other side has to be a. This is the 3'. This is the 5'. And then put the A here. So the A also has a 3' carbon and a 5' carbon, right? So the question is, can we reverse A such that the 3' carbon of the A is binding to the 3' carbon of the C? So what would that look like? So that would be, see. So the sugar, let me see if I can draw the sugar. sorry, this is one more here. And then there's the base. And this is in RNA, this would be a hydroxyl group. Let me just write it there. So it's as a reference because maybe it's easier to keep track of things. So it's the 2', this is the 2' hydroxyl, this is the 3' hydroxyl, sorry, the 3' carbon. It's the 3' there. And then this one is the 5'. Okay. So then if we want to attach, so let's call this is the A base instead of the, and then we want to attach a C so that the 5' carbon, so 3' carbon is attached there to the 3' side. So then we have to sort of write that upside down. It's difficult to do. Let's see if I can do that. So then we have in the case of RNA, we'd have the hydroxyl group there. And then we would have the nucleobase, the C. It would be here, that would be a C. And then we would have the sugar, I mean the phosphate. There's a minus charge on that oxygen, and then O, and then there. So this is a 3' bond, and they exist in nature. I mean, they can be made chemically. Their existence in nature is rare, but in principle, we can have 3' bonds, and then this would be a 5'. This would be the 5' carbon then. Anyway, maybe you can, since it's already approaching 11, maybe you can think about this, whether such structures are possible, whether you can make a tetranucleotide using, by combining 3' and 5, you can essentially changing the directionality of the atoms, and what kind of chemical diversity you get, and whether that chemical diversity is sufficient to encode amino acids. So one thing, of course, that's missing if you're having tetranucleotides is that you would have to keep some order to the tetranucleotides, right? Because so now we know that information is stored in codons, and these codons have 3 bases, right? So 3 nucleotides per codon. But DNA is a long string, so you can have 3, then another 3, and another 3, and the order is important. So you just can't randomize codons and get the same protein. So in addition to being able to encode sufficient information in each tetranucleotide, you also have to be able to organize the order of tetranucleotides in some sort of a sequence that then encodes for a protein. So anyway, this exercise is just to show you that, in fact, there is a fair amount of chemical diversity, even in relatively simple chemical structures, and that in principle, it should be possible to encode for all of the proteins, but you would still need some sort of a larger structure, like the way we write on a piece of paper. We assume that at least in not in Arabic, but in many languages, you write from left to right and then from top to bottom. And so that then, what we're writing on a piece of paper, and so then we know what the sequence of words should be. So we need a similar scheme here to be able to organize these tetranucleotides into a larger sequence, each of which would presumably each tetranucleotide would then encode a protein, and then the sequence of the tetranucleotides would have to be organized, for example, by binding on some sort of a surface or in some other macromolecular structure. Okay. But let's go on. Let's see. Okay. So anyway, so the information storage is closer to combinatorics, so how to count things. So it's good to have a little bit of a sense of how to do this. Do you know, for example, how to calculate how many codons there are based on 3 nucleobases per codon, right? So A, A would be 1 codon. How many 3-letter codons of 4 bases are there? Anybody?
T4.
Excuse me?
64.
Right, so there's 64 possible codons. Excuse me. So this is determined by the size of the alphabet, right? Which is 4. So A, C, G, or T. So that's 4 letters. And then the size of the word is, in this case, 3. So 4 to the 3rd is equal to 64. And this is a useful, very useful alphabet size, which in the case of DNA is 4. I mean, excluding non-canonical modifications like whatever, methyl, cytosine, or whatever. It's just as taking the 4 canonical bases. If we can form words of three letters of this alphabet, then we can have 64 different words. And that is then sufficient and redundant for encoding all the amino acids that we need, plus the start and stop codon and so forth. So if codons were 2-letter words, how many codons could you encode? How many proteins could you? How many amino acids could you encode?
Question.
Yeah, if there were only if the codons were two nucleosides long, how many amino acids could you encode?
16.
So it's 4 because we're staying with the same size of the alphabet, A, C, G, T, but instead of a 3, it's a 2, right? So yeah, so 16. And so keep in mind that this type of calculation is very useful. Say like how you want to know the molecular diversity of a 20-mer, right? So how many possible 20-mers? Say you want, you're designing a primer that is a 20-mer, and you want to know theoretically how many 20-mer primers are there. So before, because there's again 4 letters of our alphabet in this case, and then 20. So what is 4 to the 20th? So this is 10 to the 12th. So this is starting to be a very large number. Okay, so it's useful to keep in mind that it's good to be able to come up with a rough sense of the chemical diversity possible, even with relatively simple, chemical structures like, DNA. So even a short DNA sequence, say 5-mer, 6-mer, 10-mer, you can encode enormous information in that short molecule. Okay, I think that's the end for today. I will send you a homework. So we'll just do a few homeworks during the next 10 weeks. But I think probably today will be one day, and just try to get it back to me by the next week sometime. Okay, so send me an email if you have any questions or concerns. Otherwise, I will see you in 1 week. And thanks a lot for coming, and I appreciate your attention. Okay, bye.
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 Bye.